we are SRE-China. In our project on a small molecule of peppermint WGX50, we have found its pharmacological activity to be effective in anti-aging, as well as in preventing and slowing down the process of Alzheimer's disease. The reason why our team chose to address the global anti-aging context is due to the increasing number of elderly people. The World Health Organization has predicted that the number of older people aged 60 years and older will reach 1.4 billion by 2030, and that this number will continue to grow, reaching an estimated 2.1 billion by 2050. This problem of global aging is becoming increasingly serious and poses enormous challenges for families, public health, and the economy. Healthy aging is a real human need, as stated by the Universal Declaration of Human Rights. Therefore, development of anti-aging drugs for a long and healthy life is an important research topic. Up to now, a number of anti-aging drugs have made significant progress in preclinical research, showing significant anti-aging and life-extending pharmacological activities at the model animal level. However, to date, no anti-aging drug has been shown to be effective in clinical trials, and even the popular anti-aging drug NMN has been proven ineffective in recent clinical studies. The challenges in this area remain enormous.
As a result, our team undertook a series of human practices to address the existing research challenges. It was like a thread that ran through our project, giving us correlations along the way.
Pre-experimental studies by our IGN team, SRE, have shown that WGX50 small molecules exert significant effects in anti-skin aging. At the same time, we used model organism nematode experiments to explore the function of WGX50 small molecules, as well as computer simulation technology to simulate the interaction between WGX50 and proteins, and found that WGX50 small molecules can be bound to the target proteins. It can effectively inhibit the role of alpha-beta, that is, to prevent apoptosis protein expression and reduce the apoptosis, thus effectively inhibiting the secretion of inflammatory factors. On the other hand, it can promote intracellular collagen production and inhibit the generation of reactive oxygen radicals, thus inhibiting and exhibiting anti-aging and antioxidant effects. In our project, we have comprehensively utilized the cross-fertilization of pharmacology, molecular biology, and bioinformatics of Chinese herbal medicines in screening natural small molecules for Alzheimer's disease from a database of Chinese herbal medicines using AI and computer-assisted drug design. We also explored and verified the molecular mechanism of Alzheimer's disease treatment by combining with pharmacological and molecular biology.
For the preliminary investigation preparation of the project, we conducted in-depth interviews with physicians and specialists in medically related fields and gained reflections from them. From a physician's consideration, whether it is possible to rapidly screen drug components that are compatible with the treatment of Alzheimer's in a cost-effective yet safe and stable manner. Synthetic biology in Alzheimer's disease. Efforts are still needed to prove safety. Drug development cannot rely solely on basic experiments. Clinical trial results are the most crucial. Given the specificity of Alzheimer's disease and the anti-aging properties of WGX50 in practical application, and can be formulated into health product that prevents Alzheimer's through mechanisms such as antioxidation and anti-inflammation and anti-aging. From the consideration of the specialists, Early intervention should not be limited to combating Alzheimer's itself. Instead, it should be aimed at the public's pursuit of a healthy later life, with anti-aging serving as a precursor to preventing Alzheimer's. Interviews confirmed the correlation between anti-aging and Alzheimer's. Meanwhile, it hinted at the key of experimental research, which is to demonstrate whether WGX50 possesses efficiency in combating cellular aging, reducing oxidative stress, and fighting neuroinflammation.
In China, pepper is a common food additive and traditional herbal medicine. In people's long-term practice, the efficacy of Chinese pepper in the fields of analgesia, anti-inflammatory, antioxidant, and antibacterial has been confirmed. Studies have shown that the active ingredient of WGX50 has anti-inflammatory and antioxidant effects similar to those of Chinese pepper, and is a candidate for anti-Alzheimer's disease. As of now, there are very limited studies on the anti-aging properties of WGX50. The earliest evidence from experimental studies suggests that WGX50 inhibits aberrant calcium-ion inward flow in red neurons, and that disturbed calcium-ion regulation is a recognized symptom accompanying brain aging. In a follow-up study, WGX50 was found to inhibit galactosidase activity, downregulate malondialdehyde and reactive oxygen species, and regulate the expression of the senescence secretory phenotype molecule TGF-beta1, ultimately delaying skin aging in mass. Accordingly, we deduce that the WGX50 is likely to have systematic anti-aging pharmacological activities.
In our study, we integrated the cross-fertilization of screen natural small molecules for Alzheimer's disease by using artificial intelligence and computer-aided drug design, and then explored and verified the molecular mechanism of Alzheimer's disease by combining with molecular biology. In the view of the previously described beneficial effects of WGX50 on Alzheimer's disease, pain relief, drug-induced heart injury, and skin health, we sought to investigate its impact on longevity of C.elegans. Interestingly, WGX50 significantly prolongs the lifespan of C.elegans, and the best lifespan extension effect was observed at the concentration of 50 micromolar. The molecular force field is an abstraction of molecular systems based on the theory of classical mechanics and can be used to study biological systems and assembly pathways of different sizes and complexity. The principle of molecular dynamics stimulation is that, after obtaining the initial motion information of a microscopic particle and the interactions provided by other particles, the position of the particle at the next moment is derived by solving the equations of motion. Our project in vitro test showed that WGX50 was shown to be able to depolymerize alpha-beta aggregates in vitro and improve the memory of demented transgenic mice in an extended test. This provides a test basis for the potential of WGX50 to extend lifespan and improve cognitive behavior.
In longevity experiments, worms were allowed to grow to adulthood before exposure to any stimuli to eliminate the contribution of any developmental effects on lifespan. Embryos were synchronized and allowed to develop on NGM plates for about 3 days at 20 degrees Celsius. Adult animals were then transferred to new plates that had been coated with WGX50. Growing adult worms at 20 degrees Celsius was scored and well maintained every other day. Animals were judged as dead when they ceased pharyngeal pumping and did not respond to prodding with a palladium wire. Then, because four novel genes take no roles in WGX50-mediated longevity, owing to a combinatorial effect enabled by the drug. To decipher the mechanistic pathways behind the detected phenotypes, we performed RNA sequencing for adult worms treated with WGX50 or vehicle. And we analyzed the distribution of 434 transcriptional genes as illustrated in heat map. Next, RNA interference was conducted to investigate the unexplored role of these genes in WGX50-mediated longevity. RNAi is among the processes that use short RNAs as guides for sequence-specific silencing. Long double-stranded RNA causes sequence-specific mRNA destruction, which is known as RNAi. This phenomenon was first identified in C.elegans.
To summarize, we can derive the following results from the experiment. In accordance with our study, WGX50 extended lifespan in C.elegans, in which the dose 15 millimole per liter increased the median survival to 45% from 22 to 32 days at 20 degrees Celsius. To this end, we concluded that WGX50 prolongs lifespan in C.elegans and has anti-aging and life extension pharmacological effects. And WGX50 medicated survival increase was not diminished by RNAi any of these genes. Non-groups of F44E 5.4/5 or F59B 2.12 RNAi worms were tested to be significantly different on survival compared to the control while treated with WGX50. Thus, we concluded that these novel genes are not causative for WGX50 medicated longevity. In conclusion, synthetic biology is the interdisciplinary discipline that combines biology, engineering, computer science, and chemistry. It centers on the design and construction of new biological systems to achieve specific functions. We have successfully investigated and analyzed the ability of WGX50 molecules to efficiently regulate intracellular signaling pathways, as well as influence neuronal metabolism and protect neuronal cells from oxidative stress damage through a synthetic biological approach. This achievement marks an important step forward our understanding and utilization of natural compounds. As a result, our projects can bring social significance, as demonstrated by research on the WGX50 provides new health protection for the elderly and helps to improve their quality of life. And Alzheimer's disease and other aging-related diseases are costly to treat, so the application of WGX50 is expected to reduce the incidence of these diseases, thereby reducing the burden on the healthcare system. However, this discovery will inspire more scientists to devote themselves to synthetic biology and neuroscience research, and promote technological progress and knowledge innovation in related fields. With the deepening of WGX15 molecules research, public awareness of aging and Alzheimer's disease will be enhanced, which will help to strengthen health awareness and implementation of preventative measures.